After we understand how the tongue is organized, with the papillae, the taste bud, and the sensory cells, so what kind of taste can we sense? In fact, normally when we say the tongue can taste, we say that it can taste bitterness, sourness, saltiness, and sweetness. But there are more than 4 different tastes. So what are they? And they actually have some important implication. For example, bitterness, as we said, in fact, bitterness is caused by compounds such as phenolphthaleurea, polyphenols, which we found in a lot of plants, flavonoid, terpenes, organic bases like glycosides or so. These are usually associated with toxic substance. How about sourness? Actually, what we are sensing is the amount of hydrogen ion present in the food. So it always comes with the acids such as lactic acid, acetic acid, citric acid, like that. How about the saltiness? It's largely because of the minerals such as sodium. It's because of all this ionization of inorganic salt that gives the sense of saltiness. How about sweetness? In fact, what we are monitoring is the presence of the hydroxy group in a lot of these sugars, such as glucose or fructose, and sometimes even ethanol or glycerol. Now, the final one, in fact, is umami. I said that it's a taste of amino acid. What exactly it is? Amino acid, we say that actually, and the nucleotides, both of them can give you the sense of umami. And that is largely because of this glutamate structure. Now, these taste substances, they have one common property. Whatever it is, acid, minerals, these polyphenols, or amino acid, they are all polar. That means they carry charges. And they are water soluble, so that they can easily be dissolved in the water. And so that when we taste, we want them to be in the water as a solution so that we can perceive them. And most of them, they are relatively stable in water because they are not volatile. So they're not going to be vaporized and disappear gradually, all right? So this is the collection of taste that we're going to sense. And a lot of times when you read the textbook, you may see that, well, along the tongue, we have different regions of the tongue that they can taste different kind of bitterness, sourness, saltiness, or sweetness, and they have a taste map like that. Later on in this particular section, we're trying to go through another exercise to find out whether it's real. Now, let me talk a little bit more about the science of it. We say that in the sensory cell, in the taste bud, they have a lot of microvilli up there. What they have is that, in fact, in this particular cell, on this microvilli, they have a lot of all these receptors. And these receptors can be generally classified into 2. One of it, we call it the ion channels. Basically, you look at that, there's a channel sitting on the surface of a cell. And whenever there is any salt or hydrogen ion, they come through it. They would depolarize the cell, and therefore, it triggered the firing of this neuron. The other type of it is for the sensation of the sweetness, bitterness, and the umami. These, we call them G-protein coupled receptors. Sounds difficult. But very simply, they are simply receptors. They have very specific configuration so that they would be able to capture the bitterness molecule, the umami molecule, or the sweet molecule. And when they're activated, they can trigger the firing. How do they do that? In fact, they all come into the cell, leading to the depolarization of the cell, through a common machinery. Now, in this process, when the cells get activated, what do? They fire out signal. And this depolarization process results in the nerve, they would have their membrane potential being altered so that they're going to fire and trigger a spike with the influx of the ion, but then they quickly restore it so that this particular signal will be transmitted along the axon so that they can go from one part of the neuron to the other end of the neuron. And then through some chemical connection, they will be connected to other neurons in the brain. Now, in this process, in fact, we need to ask ourselves, are there parts of our tongue which are more sensitive to a particular flavor? Or all parts of the tongue, they are equally sensitive to the same flavor? Now, this it is quite a bit of dispute because it varies from people to people. So let me put it into some of the examples. For example, we say that, well, the taste bud, we can sense the sodium chloride only. Or actually, we can sense both the sodium chloride and the sucrose. We find that, in fact, within the same taste bud, there are different sensory cells. Some of the sensory cells, in fact, they can sense sodium chloride. But some of the sensory cells, in fact, they can also sense sucrose. So therefore, what it means is that within one taste bud, in fact, they can sense both sodium chloride and the sucrose. Now, but then the question is that, what's the composition of all the sensory cells in this taste bud? Sometimes they have more sensory cells which sense one. Sometimes they have more sensory cells sense the other. So therefore, that would result in different kind of sensitivity. A second part of it, we say that the bitter receptor, in fact, they are expressed in a subset of the taste cell in all the papillae on the tongue. This is done in the experiment in the rats. But however, what we find is that these receptors, they are more densely populated at the side and the back of the tongue. So therefore, usually people would say, well, when I sense bitterness, usually it's coming from the back of your tongue or the side of your tongue. Now, this tells you that the taste map, again, it varies. If you look at the sensitivity of our tongue to all the tastes, they are distributed across the entire tongue in different regions. So what we need to consider is that when we say that we are sensitive to a particular taste, we need to question whether it is because of the density of the papillae, the density of the sensory cell, and also how the sensitivity is related to the number and amount of receptors available in that particular region. Now, very easily, we can use an example as an illustration. We say that when we sense sugar, we are sensing glucose. And in fact, the sensation of sugar is triggered by a receptor. We call it T1R3. This particular receptor is a G protein coupled receptor, a receptor. It is present in our tongue. Are they evenly distributed? The answer is no, they're not evenly distributed. They're more heavily focused on the tip of your tongue. But the question is that, can different regions of our tongue sense the sweetness? The answer is yes. Because, in fact, we find that this receptor is not just present in the tongue. In our entire intestinal tract, in fact, there are also this receptor present. So, you recall that we talk about sometimes we want to sense fullness after eating some meal. And for experiments that we do with animals, we can inject glucose into their duodenum. And in fact, they can still feel the fullness. So somehow that must be a relationship of that. So in the real life, when we look at this particular receptor, look at where they're expressed, in fact, we find that they are expressed in the taste bud. So that means your taste bud have the receptor to sense the sweetness. But it is more than that. We see that it's present in the nasal epithelial cell. Well, somehow in your nasal cavity. So why are they there? We have this receptor in the trachea near the lung, and we have it in the stomach, we have it in the bile duct, and we have it in the intestine. So basically what it says is that, in fact, these receptors, they are not just there for us to perceive the taste. They probably would have additional function elsewhere along the intestinal tract so that allow them to monitor the presence of glucose somewhere in the body.